Crinoid time! All right, here are some crinoids. Now, crinoids usually, so they're stalked organisms generally, and they mostly are sessile, so they're going to be rooted to the bottom. Sometimes they'll even have a holdfast where they kind of grow over a piece of rock or something like that. There are some kinds, though, including some modern ones that we can see up here, that are actually at least have the potential to move themselves around. So all those arms on a crinoid, those pinnules down the side of the crinoid, potentially can be controlled and can work kind of like limbs. So think of the cup. That's where the mouth is. So think of that as like the head. And the brain is actually a ring that's going to run around this, kind of just underneath the surface. Now it has an anus coming out of the center of its head, but don't worry about that. That's a crinoid thing. So think of this as kind of like a skull. Imagine that as being kind of like a spine. And so this is the thing, it's like a little skull and spine that's dragging itself along the very deep water bottom. Yeah, kind of neat. Anyway, enough for analogies. These are crinoids, and here you can see that crinoids are going to have what you could call a stem. Columnals are how they describe each of these little tiny units that makes up the stem-like part. Here we have the arms, and so the arms are going to essentially filter feed. They will catch little particles, little organisms, and they will move them on little sticky, basically suction cup feet toward the center. So they're sort of spreading wide to catch as many things as possible, and then moving all of those down and into the mouth, basically. There are many of them, particularly from the late Paleozoic, that develop an extended, it's called an anal chimney. And so here's the anal chimney. And when they have one of those, they'll have a ring of spines around the top. They'll be sort of flattened spines. They'll look like this. And those will be called the anal spines. And you'll see a lot of those in the fossils from the Pennsylvanian in particular. And then there will be these more sort of conical spines with a triangular base as opposed to kind of a pentagon, sort of flat canoe paddle looking thing. Those are going to be the oral spines, and so those go around the mouth. So yeah, let's talk crinoids ecology. Crinoids tended to be found in lagoon environments, although then as now there were specialists for different environments. Crinoids are believed to be highly ecologically tiered, which means they would grow to a certain height so that they can filter feed all of the little particles at that height. And so each species would dominate a particular height, just as there are different trees that dominated different heights in the forest canopy. There are going to be different crinoids that do that on the lagoon, on this little sort of shoal. They're not particularly well put together. They do not do well in high energy environments. So you would tend to find their fossils behind something like a reef, which is what we mean by a lagoon. And yeah, you can see all the little creatures that are found with them. And yeah, here they are. So let's look. All right. There are some crinoids. Yeah, let's move this over a little bit. They're going to build up attachments, and the attachments are just going to be modified columnal segments, and they are going to then sort of wrap around the ground. Now, like in other echinoderms, the crinoid body parts, those mineralized parts, are going to be made of stereom, which is a calcium carbonate. And it's kind of a light, bubbly calcium carbonate compared to more solid calcium carbonate, like you might see in an oyster shell or something. But they are quite strong in the same way that a honeycomb design can use very little material but make a very strong organism. And they don't have all the same sort of advantages that something like a coral would have in terms of building the actual skeleton. So yeah, they're doing the best they can with what they've got. And so here are the little modified columnal segments that are going to then sort of wrap around whatever's nearby, including this little coral. Just wrap it up. There are other crinoid root-like structures that are actually going to branch. It's going to be something like a crinoid cup structure, only they're going to be modified pinnules, like the little side branches that would come off. It's going to develop a bunch of those, and then you're going to get branching on the branches as well for some of these. And they spread out, and honestly, they look a lot like a tree root, and they would have the same function in terms of keeping the organism upright and anchored. They would not presumably have the same function in terms of delivering nutrients. That's still going to be via the mouth, which is at the top. And we're talking about the bottom of the crinoid. There are some of these things that are thought to be potentially snap coiling. So they would have little sort of brushes of these pinnules. And the idea was that they may be able to sort of extend, and if they saw a predator, they could then sort of coil back up inside these branches. You will find them fossilized like this and surrounded by these little branchy structures. So that's possible. They were able to do this, and actually kind of neat if it is correct. Some of the crinoids will have nodal grapnels, which are like little gripping devices that are going to appear at different nodes of the stalk. Yeah, and so here they're showing that there's a certain number of intermediate little segments in between each one of these grapnels. But they're quite broad, and they're kind of like a grappling hook for holding it in place. There are others that will have basically a grappling hook, cirro grapnels. So they're using cirri, the little side pieces, to make something like a literal hook that you would see, a James Bond or a Tom Cruise type toss onto the root of a building. And the idea was that this is going to act like an anchor. The crinoid can continue to grow it. So crinoids, like us, are going to have a mesodermal skeleton, which means they have tissues on the inside of that, and they have tissues that coat the outside of that. And so they can grow their skeleton from the inside because it is actually surrounded. It is sandwiched by tissue. Yeah. And these little holes that you'll see running through crinoids, that's where the endosomal stuff is. So that's, that's where nutrients are going to be transferred through from the mouth, and we're going to keep all the rest of these segments fed, basically. Fed and alive. Yeah. Here's an interesting one that developed these sort of grapnels, but only on the base. And it's thought that these were like a little cup that would sort of keep the crinoid. It would be able to sort of tip over, but then it would self-right, just like a little weeble. And so here you can see some of the assorted parts from these that were all deposited together. There's some that had a sort of a curly, it kind of looks like a caterpillar. And it's not really a root. This is just a modified part of the stem, but it would wrap around something. And may also develop little cirri on the side that would hold on to that. They look like little caterpillar legs, but they're just modified cirri, the little arms, like you can see on the side of our little walking crinoid down here on the bottom. That's not common for crinoids, like stalked crinoids, to do this, but it does happen. Yeah. Okay. There are also some that are believed to have had little anchors where they'd be able to move it. They would have like change position, and they would sort of break off one part and then continue to grow as the sediment built up. There are others that, like here's Paragacia crinus. These are believed to be maybe pelagic, or maybe they would sort of sit on the bottom, but had the ability to move. Something like a modern feather star. Here's a feather star. And if you've ever seen those swimming, they have a really neat, it's kind of an up and down motion. They sort of alternate which arms are pushing up and down. They're just the most beautiful things. But these are all crinoids. Yeah, feather stars are crinoids, and stalked crinoids are also crinoids. And so this is a very early branch, not directly related to the modern crinoids that lack stalks. But doing the same thing, convergent evolution. So If you're lucky enough to have a crinoid calyx, a crinoid cup in front of you, what do you do with this? How do you understand it? Well, we can start by numbering and labeling the different parts. So if you hold it up and you're looking at the bottom where the stem comes in, the stem will actually border onto 5 plates. You're going to see 5-fold symmetry a lot in a crinoid, but it's not quite 5-fold symmetry. It's kind of messed up 5-fold symmetry that's really 2-fold symmetry, but sometimes it's not even 2-fold symmetry. Yeah. They're contradictory little creatures. The lowest level, these are called the infrabasals because they're lower than the regular basals. And the next up, these are the basal plates. And so they're usually going to be pentagon shaped. They'll have this particular shape. And then sitting on those, articulating on top, are going to be the radial plates. And they'll also have a pentagon shape, but they'll have a big flat top. And the flat top will have grooves in it where you can put arms, basically, where the arms are going to articulate. At one point, they're going to have a plate that's called the X plate, and that's going to mark where the anus is going to be. It's closest to the anus, and so that is one part of it that is going to be asymmetrical. But this is the organization you will very commonly see in Paleozoic crinoids, and so that's why it is worth figuring out. Yeah. So the radial plates are going to have, notches and grooves, and that is where they articulate to each other. It's also where the different ligaments and muscles that would have moved each of these parts are going to be found. That's what we're looking at. That was the inside of a radial plate. When you are classifying, categorizing crinoids, you can look at the cup. So the cup will tell you what it is for sure. The stem may tell you, but we have, just as in plants, separate names for the columnal segments, which are not the same as the name for the organism. The name for the cup is the name for the organism, so to speak, because then you've at least got it, and you know for sure who you're dealing with. All the important parts are there. But yeah, you can look at their shape. So is it cone-shaped or cup-shaped or like champagne glass-shaped? Is it going to flare out in? Is the stem going to be pushed up inside so that the infrabasals actually are curved like this? Or is the stem going to form the bottom point and the infrabasals curve up around it? Or is it very flat right where the infrabasals are, and then the basals and radials are going to come up and do all the work? Those are the differences between different kinds of crinoids. And so here, break this down. What have we got here? 5 little infrabasals. Who sits on top of the infrabasals? Why, the basals, of course. Who sits on top of the basals? These little sort of like little diamonds, but we shaved off the bottom. It's like a pentagon on the bottom. These are going to be the radials, 1, 2, 3, 4, 5. So 5 to 5 to 5. The infrabasals are often fused together. It gives it more structural stability. And they're all contacting the same top segment of the stem, so that makes sense. The radials will have the arms docked on there, and there are many different kinds of arms. But what you see there is actually going to be analogous to the way we have one bone here, and then 2 bones here, and then you get all your little phalanges and carpals and metacarpals, whatever. And so think of that as kind of like a humerus, radius ulna, and then just phalanges for days. Yeah, something like that. It's analogous. It's not the same. We're not directly related that closely, but we are both deuterostomes. Yeah. All right. So that's been crinoids, and we'll have a lot of fun going through the crinoids and trying to spot all the different plates. Oh, and the extra little plate, which will often stick up at the side of the cup, that's going to be the X plate or the anal plate. Yeah. So remember that one. Know how to spot it. Yeah, if you see a crinoid cup from the side, there it is sticking up. You know that's the anal plate. Yeah. It sticks up there. It's just because there aren't any feeding arms directly attached onto that. They're sort of slotted around it. They decorate the rest of the cup. So it is pretty close to bilateral symmetry, even though it looks like 5-fold symmetry. All right. Thanks very much.